Good day to you from ChemHelp ASAP. Temperature can affect the position of an equilibrium. So a common question is whether drug discovery scientists worry about the temperature of different assays. Because drugs act in the human body, it is reasonable to think that all drug-related assays, whether in vivo or in vitro, should be performed at the temperature of the human body. So human body temperature is approximately 37 degrees Celsius. The body temperatures of animal species typically encountered in efficacy studies are also close to 37. In contrast, chemistry-based studies are very often reported at STP, standard temperature and pressure, with temperature being 25 degrees Celsius. Since equilibria, such as drug-target binding, are affected by temperature, should we be concerned about temperature differences between studies? Said another way, will ignoring the effects of temperature lead to systematic error in our studies as we move from in vitro assays to in vivo studies? Let's look more at our binding equilibrium. Here is the dissociation of a drug-target complex to unbound drug and unbound target. Dissociation is the process going from left to right. Association is the process going from right to left. In the middle is the dissociation equilibrium constant, Kd, for our reaction, the product of the product concentrations over the concentration of the drug-target complex. At the bottom is a relationship between free energy of binding and Kd. There is a temperature term in here. Kd does indeed change with temperature. Great, so we should carefully monitor temperature. Yes, we should monitor temperature just like all the other variables in the assays and studies, but a 10-degree difference in temperature will likely not make a meaningful impact on a measured Kd value. Other sources of error are likely much larger than errors from temperature. Am I saying that temperature does not matter? No, I'm saying that fluctuations in temperature are not likely to meaningfully affect the outcome of an assay. So what are the typical temperatures for assays and studies? Biochemical assays, such as enzyme inhibition assays, are often reported as room temperature. Depending on the lab, that could be 20 to 25 degrees Celsius. Binding assays, which more strictly monitor temperature changes, will report the exact temperature of the experiment, which will likely be close to room temperature. Cellular assays are performed at temperatures that are ideal for the particular cells. Bacteria thrive under various conditions, anywhere from room temperature up to about 40 degrees Celsius. Assays using mammalian cells are normally performed at 37 degrees Celsius. Temperatures do vary across these studies and assays, but it would be very uncommon for differences in results across different studies to be explained by temperature effects. For example, if a compound shows promising binding in a biochemical assay and then you tested it in a cellular assay, the temperature difference between those two would not lead to the compound being active or inactive. In summary, temperature is important to monitor, but it is not typically a determining factor in a study. Thank you for watching all the way to the end. If you have questions, please share them in the comments section. Take care.